Imagine that you're tied to a tree and bugs are crawling all over you, fire ants and animals biting you, birds pecking at you. Well, that's unlikely to ever happen to you, but this is what the tree faces every single day. How can a live being that's unable to run away defend itself? The answer: poisons.
In this episode, we discuss how remedies from apple tree bark and potions brewed from French lilac traveled from medieval folklore into modern medicine, and how discoveries in radioactivity explain why the so-called miracle drug insulin does not work for everyone with diabetes. We recount the fascinating story of how a French army doctor championed a drug once feared, and in doing so, changed the course of diabetes treatment forever.
And a German doctor experimenting with animals thought he had caused diabetes, but had in fact discovered a radical new method to control the disease.
Join us as we explore how human ingenuity co-opted plant poisons to make blockbuster medicines for diabetes and ask an even bolder question. Could the very drugs that transform diabetes care one day help extend human life itself? Welcome to the Discovery Dialogues podcast, where we take a deep dive into the history of groundbreaking discoveries in the life sciences. I'm your host, Ani Deshpande. Each episode, we will uncover some incredible, insightful, and often just insane stories behind breakthroughs in medicine. Join us as we journey through the twists and turns of scientific history, one breakthrough at a time.
I'm Pam Itkin-Ansari, a diabetes and pancreatic cancer scientist at the Sanford Burnham Prebys Institute in California.
And I'm Ani Deshpande, a cancer researcher also at Sanford Burnham Prebys. If you've been enjoying our podcast, please help us spread the word and press the like and subscribe buttons, because this is how the algorithms spread the content. Pam, in our past episodes, we've been discussing how insulin has been a miracle for diabetic patients. Then why doesn't insulin benefit every patient with diabetes?
That's a great question. And actually, at first, doctors assumed that it would. It was soon realized that for many patients, insulin treatments did nothing at all.
Exactly. Not all diabetic patients are the same. The answer is obvious to us now that there's two types of diabetes, type 1 and type 2 diabetes, but this wasn't known until relatively recently. Well, as we had mentioned, there were detailed descriptions of diabetes in many ancient cultures millennia ago, and that the disease was called honey urine or madhumeha, which is exactly what the modern name diabetes mellitus means.
Well, interestingly, ancient Indian medical texts had noted that people with madhumeha could be divided into two distinct groups.
Exactly. And the one was called Sahaj Madhumeha, which affected younger, leaner patients and had a severe disease progression. And they described another kind, which was interestingly called Apathyajanak Madhumeha. This mostly affected older, overweight patients and had a comparatively milder progression. Amazingly, they noted that this second form of diabetes usually affected the relatively affluent, well-to-do individuals.
Ani, isn't it interesting that in modern times, this sounds uncannily like type 1 and type 2 diabetes, already noticed millennia before the distinction was clearly made?
Yes, and this was a remarkable clinical insight for that time, even though there was no knowledge of insulin or ways to accurately measure blood sugar at that time.
Right. And now fast forward to 1936, when the British physician Harold Himsworth performed an experiment that would establish the molecular underpinnings of these two types of diabetes. Himsworth had a young and an old diabetic patient eat sugar, and then he injected both of them with insulin. Now, when you inject insulin, your blood sugar should come down immediately. And this is exactly what happened in the young patient. But remarkably, in the older patient, insulin injection did nothing. The blood sugar stayed high.
So what Himsworth had discovered was that there was one type of diabetes that he called the insulin-sensitive diabetes, and another that was called insulin-insensitive diabetes. Why was this the case?
Well, even though physicians could see that sugar was high in both types of patients, nobody could measure insulin yet.
Well, from the 1920s to the 1960s, Three Nobel-winning women completed key pieces of this puzzle. Marie Curie, the pioneering physicist-chemist, showed that radioactivity could be used to detect extremely tiny amounts of substances. Then Gertie Cori, a biochemist, uncovered how insulin lowers blood sugar. And finally, Rosalind Yalow, the New York biophysicist, turned these insights into developing a highly sensitive test to measure trace hormones like insulin from just a drop of blood.
Right. And that led to the big surprise. In fact, she found that many diabetic patients had no insulin deficiency at all.
That's amazing.
That's why insulin injections didn't help lower their blood sugar in Himsworth's experiments.
Pam, just to clarify,
Yeah.
type 1 diabetes means that the body makes little or no insulin
Right.
and usually shows up in younger people.
Mhm.
Type 2 diabetes, on the other hand, is typically tied to overnutrition, to obesity, where insulin is present but doesn't work really well. Is that right?
Pretty much. It used to be a clear distinction. If a child had diabetes, it was almost always type 1, and it was even called juvenile diabetes. But today, because of widespread childhood obesity, driven by diet that we're eating in modern times, we're seeing more and more children develop type 2 diabetes, and that shift has become a major global health concern. Now let's come to the older people with diabetes that we call insulin resistant. Well, it turns out if the diabetes remains uncontrolled for long enough, those insulin producing cells eventually wear out. And then just like type 1 diabetes, the patients become insulin dependent.
Oh, that explains why some older people I know with type 2 diabetes have to rely on insulin.
Exactly. So what can we do for the people whose diabetes cannot be fixed by insulin? They need medicines to lower their blood sugar. And where do these medicines come from?
Well, when faced with this problem, we do what we've been doing for centuries.
Mhm.
We go to nature to seek answers. And it was from nature, specifically from plant poisons, that we got two of the biggest blockbuster medicines for diabetes. Should we talk plant poisons, Pam?
Oh, yes. Let's talk plant poisons.
Every organism on Earth takes great pains to ensure survival, and this is also true for plants. The only problem is that even though they don't want to be eaten by a random insect or an animal or even a human being, they cannot just run away. In fact, they can't even bite or fight back with claws or fangs. They can't even hiss or growl menacingly like animals to ward off their attackers. So how do they survive? Well, as I like to put it, if you cannot rely on physics in the sense of mechanical forces to defend yourself, then you better have kick-ass chemistry.
Absolutely. Over millions of years, plants have evolved into master chemists, producing an astonishing array of chemical poisons designed to defend themselves. And we have evolved means to fight off or even savor these poisons. To drive home the point of how incredible plant poisons are, we decided to ask the vascular surgeon and amazing writer Ambarish Satwik to share what he wrote about the arms race between plants and humans that is almost poetic in its beauty. It's titled, What the Salad Tried to Do to Us.
Here's Ambarish from New Delhi, India.
What the salad tried to do to us. The first ambition of any plant is to make itself uneatable. Long before anything clever walked on earth, plants settled on poisoning anything with a mouth. If you can't run, you better taste terrible. The only way to stay alive was to face your predator, dinosaur, beetle, goat, man, with chemistry. Plants got clever in the only way available to them. They made toxins, astringents, enzymes, fibrous defenses, which made them and their parts bitter, also indigestible and outright poisonous. In evolutionary terms, it was a cunning move. Not that the plant knows anything about cunning, but the natural consequence was deterrence. You bite, you suffer. You learn, you stop biting. Humans responded in the human way, by using fire and other things. We boiled, we burned, we soaked, we crushed, we fermented, we pickled. We found ways to take the bitter poisonous offerings of plants to our hungers. We hacked our own biochemistry, made over 50 types of cytochrome P450 enzymes. They're fascinating. A plant toxin molecule is like a greasy stain. Cytochrome P450s act like detergent. They grab it, break it apart, make it dissolve in water, and wash it away. Cats don't have a lot of this stuff. The thiosulfates in a couple of cloves of garlic can trigger a dangerous oxidative crisis in cat blood. About 4 cloves of garlic can be a potentially lethal dose. A tiny amount of solanine from a raw potato can sicken and kill a rabbit. Without a particular kind of P450, a potato would be a last meal. We figured out how to eat the enemy, and then we got kinky about it. Our tongues learned to relish the sting and the rot. We started liking the burn and the bitterness. We started having poison for the plot. Hot sauce, tannins, coffee so strong it peels paint. Flavor is a toxin we've come to applaud. Plants spent 100 million years trying to kill us. We've spent 100 million years learning to savor their attempts. That's the story, give or take a few famines and a few million deaths.
Wow.
Yeah, that was beautiful. And you know, Ani, in fact, using poisons from nature as medications is not new. It's not even unique to humans. In the animal kingdom, there are several instances of animals using plants as medicines.
There's evidence that African elephants eat a plant from the borage family in order to help induce labor.
You know, Ani, if my pregnancy lasted 22 months like an elephant, I would have desperately searched for a plant that could help me as well.
That's funny.
And humans have been doing this for centuries, maybe even millennia. One of the most famous cases is of Otzi the Iceman, a mummified body from 5,300 years ago that was found frozen in the Alps. What was fascinating is that scientists found that not only was he infected with a parasitic worm, but tied to his clothing were mushrooms that were known to have activity against these worms.
And this is perhaps the earliest evidence that humans were practicing self-medication long before recorded history. I would like to recommend a beautiful book called The Most Delicious Poison by Noah Whiteman for those who are interested in this topic.
It's wonderful.
And I hope we have now set the stage to talk about how a plant poison, an extract from the bark of the apple tree, transformed diabetes treatment.
Few modern diabetes drugs have had an impact as profound as the SGLT2 medicines taken by millions. And their story begins with a plant poison hidden in apple tree bark and an observation made more than a century ago.
Pam, I find it fascinating that people would even test tree barks. I mean, of all the parts of a tree, the bark sounds the least appetizing.
I know, but in fact, willow bark extracts were used to treat fevers and pain for thousands of years. And now we know it had salicin in it, the chemical foundation for aspirin. So if you've ever taken aspirin, you've already benefited from drugs inspired by tree bark extracts.
And here's another surprise for people who love gin and tonic. For centuries, indigenous cultures of the Amazon used the bark of the cinchona tree to treat fevers. And when European colonizers learned about it, they spread the cinchona tree across their colonies to fight malaria.
Right.
And when French chemists isolated the active ingredient, quinine, it was used to save hundreds of thousands of lives. But it also enabled colonial expansion by making tropical regions safer for the European settlers and empires. To make the bitter quinine drinkable as a daily prophylactic, the British added sweet gin, and that's what created the gin and tonic that we drink even today.
Oh, so they didn't invent gin and tonic to relax. They invented it to survive.
In fact, Winston Churchill once said, and I quote, "The gin and tonic has saved more Englishmen lives and minds than all the doctors in the empire." And this just reflects how profoundly this bitter plant extract shaped survival and morale in the everyday rituals of the empire in the age of malaria.
Isn't that cool? So Pam, now let's discuss how plant poisons inspired two of the biggest blockbuster drugs for diabetes.
Yes. In the late 19th century, Joseph von Mering, the German scientist who had first shown that the pancreas was the organ important for diabetes, remember in our first episode, He was testing a number of folk remedies, including plant extracts in dogs. When he gave them a substance called fluorescein extracted from apple tree bark, he found something shocking. He saw that this compound was increasing sugar in the urine. And so he was really concerned that this drug would cause diabetes.
In fact, I read that he called it in German Künstliche diabetes, which means artificial diabetes.
Oh, and I also love how you use every opportunity to use your German.
Yes. All right. So to understand what was going on here, we have to know a little about what our kidneys do.
Right.
Every day, our kidneys filter the equivalent of a bathtub of blood, making them one of the most efficient filters ever. And most people know that one of the main roles of the kidney is to flush out waste from our bodies.
Mhm.
But at the same time, it has to save important things like sugar, vitamins, and key minerals.
Yeah. Remember that all our ancestors lived in times when food was scarce. So this is a way the bodies evolved to retain every bit of sugar that's consumed. Now, for diabetic patients who have too much sugar, these exciting experiments with apple tree bark had revealed a new pathway to reduce blood sugar by allowing the kidney to spill sugar into the urine.
I will pause for a moment to repeat this. The kidney saves sugar. And what the apple tree bark extract was doing was that it was preventing the kidneys from returning the sugar to the blood. Plant medicines are really awesome, right? So why not just eat the plant or use plant extracts? We could cure all of our diseases, as some of the influencers on the internet seem to say, isn't it?
It's true.
Well, if curing diabetes were as easy as chewing bark, we would have solved this centuries ago. In fact, many human medicines, even the modern ones, are derived but then modified from plant products. If we think of aspirin, metformin, quinine, morphine, Taxol, vincristine, and the list just goes on, they only become approved medicines once chemists have isolated the actual substance from the plant, the
Mhm.
active ingredient
Right.
that is responsible for the beneficial effects. And then they often will modify the chemical to reduce its toxicity and improve its potency and stability.
And this is exactly what modern chemists did with the apple tree bark extract. The natural compound was quickly destroyed in the gut. It messed with sugar transport everywhere and had toxic side effects. So chemists made new versions of it. And the result was a brand new drug family, the flozins, known popularly as SGLT2 inhibitors. These drugs have fundamentally changed diabetes care. They provide durable glucose-lowering effects, as well as protection for the heart and kidneys. And today they're taken by tens of millions of people worldwide.
Pam, I have so much admiration for Joseph von Mering. He was really not an ordinary scientist. In fact, when he died in 1908, he was honored for his many achievements. He had proven that the pancreas was central to diabetes. He had pioneered barbiturates.
Wow.
And interestingly, he is also the developer of paracetamol. No one could have imagined that more than 100 years later, that forgotten compound from the apple tree extract would inspire one of the most powerful and widely prescribed classes of diabetes drugs in the world. So now let's talk about how another plant poison from an aggressively growing plant designated as a noxious weed in many states in the United States inspired a drug that is taken every day by nearly 150 million people.
Wow.
It's more than the populations combined of Germany and France. And this is the story of metformin that begins in medieval European folklore.
There's a plant called the French lilac that grows in Europe, the Middle East, and in Northern Africa. For centuries, extracts of this plant were used to increase milk production in lactating women, and also for a variety of disorders, including frequent urination and thirst, the classic symptoms of what we now recognize as diabetes. In fact, first described in Nicholas Culpeper's Complete Herbal, published in the 17th century, it was used right up until the 1930s for treating symptoms of diabetes with no understanding of the chemistry involved.
In the early 20th century, scientists figured out that the extracts from the French lilac were a rich source of a compound which lowered blood sugar, but it was too toxic to be used directly as a medication, and significant efforts to improve this drug were needed.
But then history intervened. The Second World War had begun, and chronic diseases like diabetes took a back seat to infectious diseases, that were killing soldiers across the globe. European soldiers fighting in Asia and Africa were highly vulnerable to diseases like malaria and influenza, creating an enormous demand for medicines such as quinine, which came from the cinchona tree bark. But there was a problem. Drugs made from plants depend on access to the places where those plants grow.
Exactly.
And during World War II, Japanese forces cut off Allied access to Southeast Asia, the primary source of quinine, triggering a medical emergency.
So faced with this medical crisis, the Allies were forced to rethink how medicines were made. And these wartime programs reinvigorated a radical idea that natural medicines could be rebuilt and improved in the laboratory. This freed drug discovery from the limits of geography, scarcity, and toxicity. This all-out wartime effort brought together chemists, biologists, and physicians to invent synthetic chemical alternatives.
Yep.
An emergency program that actually became one of the largest drug development efforts of the 20th century.
Yeah, scientists threw the kitchen sink at the problem of these infectious diseases, testing tens of thousands of compounds for efficacy against malaria or flu. One of the scientists found that a chemical compound, like the one from French lilac, proved useful to treat flu. And interestingly, he speculated it might be effective because of its ability to lower blood glucose. And that made the body less welcoming to the flu virus. And this paper in a Philippine journal attracted the attention of one of the most important characters in the history of metformin, the French physician Jean Stern.
Jean Stern was an extraordinary man. As Clifford Bailey writes in the journal Diabetologica, Stern was an army medic, and during the World War II, he was captured and imprisoned. Somehow Stern managed to escape to Morocco, which was then a French colony, and there he worked as a musician. He would later return to France and even participate in the liberation of Toulouse.
Quite a person.
But despite these heroics, Stern's greatest achievements would be the development of metformin as a drug for diabetes. In 1957, Stern published a paper as the sole author in a French language Moroccan journal describing his findings with a drug that had almost been discarded.
In this obscure paper, now considered perhaps one of the most influential in the field of diabetes, Stern showed that metformin was highly effective at lowering sugar in many animal models. And then he showed it in human patients. And importantly, in stark contrast to its highly toxic chemical cousins, metformin had an incredible safety profile. And in the paper, Jean Stern called this drug glucophage, which meant the sugar eater. And this went on to become a first-line therapy for many patients with diabetes. In fact, it's still sold by Stern's name glucophage, and approximately 80 million kilograms of metformin are consumed worldwide every single year.
Even Jean Stern could never have imagined in his wildest dreams just how powerful this drug would become, not only in treating diabetes, but in revealing benefits that reach beyond it.
Now, to understand why metformin may have benefits beyond diabetes, we need to talk about prediabetes. Remember, more than 500 million people around the world live with type 2 diabetes.
And importantly, diabetes rarely appears out of nowhere. Type 2 diabetes usually builds slowly over time. In fact, long before blood sugar crosses the diagnostic threshold of a hemoglobin A1C value of 6.5, the body is already struggling, and that intermediate state has a name. It's called prediabetes.
And if we look at the sheer number of people with prediabetes, this is estimated to be 10% of the world's population.
Wow.
And worse, prediabetes, often undiagnosed, generally leads to type 2 diabetes within the next 10 years. So medical professionals see an avalanche coming, and they need to intervene somehow.
So can you actually prevent the progression of prediabetes to diabetes? For this podcast, who better to ask these questions than the world's leading expert in clinical trials for the prevention of diabetes? In fact, we have on our podcast Dr. David Nathan from the Massachusetts General Hospital. If you have ever been tested for hemoglobin A1C levels, it was Dr. Nathan's work that paved the way for the adoption of HbA1c standards, all those numbers and cutoffs for diagnosing diabetes and prediabetes.
In fact, Dr. Nathan has led some of the largest and most consequential clinical trials in diabetes. And here's what he had to say about pre-diabetes.
And so what is prediabetes? It's not a disease in
Yeah.
the sense that we don't think that it is accompanied by end organ damage. It is the prep school, if you will, for diabetes,
Yeah.
because people with it are at far greater risk to develop diabetes.
In the 1990s, David Nathan asked the important question, what can we do to prevent progression to diabetes?
Many, many years ago,
Yeah.
started in the early to mid 90s. And it seemed clear at that point that the diabetes epidemic was upon us. And maybe we were prescient in recognizing that diabetes was going to become even a bigger epidemic than it was. So, of course, I'd been using metformin as a clinician for a long time. But when it came down to looking at this diabetes epidemic, which was clearly born of our epidemic of obesity and overweight in this country and around the world, where people were really much less active than they had been, were gaining weight. And lo and behold, turns out that type 2 diabetes is the natural consequence of that. People who are, really obese to the point of being at the what's called BMI greater than 35,
Yeah.
which used to be a small minority, but which will soon represent something like 40 to 50% of our country.
I know.
The risk of diabetes for that group compared to someone with a BMI of 22, a thin, otherwise healthy person, it's going on 50 to 100-fold more.
Ugh.
So if our continent doesn't sink under our collective weight, that doesn't happen first. Unfortunately, diabetes will become even more popular. In 1993, when it was clear that there was a public health need to try to prevent type 2 diabetes,
Yeah.
I was lucky enough, they called on me and asked me if I would help lead it. And that study used metformin.
Now, in this study, one group started with lifestyle interventions, the kind your spouse tells you to do. Lose weight and do some exercise.
Just get off the couch.
We had two approaches. One of them was, not surprisingly, what we called an intensive lifestyle approach,
Mhm.
which was to try to get them to lose a certain amount of weight. We were aiming for just 7%
Right.
loss of
right,
body weight. They
right.
all start off, as I said, overweight or obese, and also increase their physical activity levels.
As
to
I remember, 150 minutes.
150
a
minutes
week
a week, right?
of brisk walking or
Walking.
something.
Yeah, it wasn't, they didn't have to do kung fu. They didn't have to go out and play competitive, I
Yeah.
don't know, cricket
No.
or something. All they had to do was walk, which is what most of our participants did.
Yeah.
And that intervention reduced the risk by 58%.
Yeah. It's
And that was
like-
huge.
It was this study that showed what we all know now, that lifestyle interventions like diet and exercise can prevent diabetes. But can a drug accomplish this?
The other intervention that we used was metformin.
Mhm.
We chose metformin because it was well studied. It was an old medication even at that point. It was incredibly inexpensive. We selected it because it was an incredibly safe drug. It was well tolerated,
Yeah.
and it did the things we wanted.
Yeah. Yeah.
Increased insulin resistance and might secondarily improve insulin secretion. And although it didn't lower the risk as much as the lifestyle did, it did lower the risk by 31%.
And as this important trial was ongoing, Dr. Nathan got an ominous sounding phone call.
The independent monitoring board was looking at the data as it came in, and I got a call one day and said, we want you to stop the study.
Yeah.
And,
I remember it's in the paper
yeah.
that it was ended early
It
because
was ended a year early because of, now sometimes when you're an investigator, you go, you get that call and you go, oh my God,
this is bad
there's
news.
toxicity, there's something
Right.
bad.
No,
But this was because we had demonstrated that we could decrease the development of diabetes. And they said, look, you've shown what you said you would, you got to stop the study.
And the success of this trial cemented metformin as one of the first therapies proven to reduce the risk of progressing from prediabetes to type 2 diabetes. But we are not done yet with this drug. Metformin may also influence fundamental processes of aging, placing it now at the center of a new conversation about longevity and health span.
and it's worth noting that the risk of virtually every major disease that we fear, like Alzheimer's, osteoporosis, cardiovascular disease, and stroke, increases steadily with age.
Now, it may seem outlandish, but could metformin help alleviate these conditions? Because hundreds of millions of people had been taking this drug for decades, scientists could ask a simple question. They could just look back in time at people who took metformin for decades and compare them with similar people who did not, and that could give them a hint of whether metformin changed the rate or severity of these diseases. And the data looked promising.
Over and over again, metformin users were found to have lower rates and often milder forms of these age-associated diseases when compared with similar people not taking the drug. Even though this didn't prove cause and effect, the data were compelling enough to warrant the launch of rigorous, carefully controlled clinical trials to test whether metformin might truly target the biology of aging itself.
And the clinical trial that will ask this question in a systematic manner is called the Targeting Aging with Metformin trial, also called the TAME trial.
For our episode, we interviewed Dr. Nir Barzilai, the director of the Institute for Aging Research at Albert Einstein College of Medicine, and who also leads the Glenn Center for the Biology of Human Aging Research and NIH's Nathan Schock Centers of Excellence in Basic Biology of Aging.
Nir Barzilai has spent decades studying the biology of aging, particularly why some people live long, healthy lives with remarkably low rates of age-related disease, and has been a leading advocate for testing whether existing drugs can target aging itself.
I'm a diabetologist. A lot of my research in the beginning of the years were related to diabetes. I'm still seeing patients. But I was always interested in aging. Long story, but when I went to medical school, I learned that you can measure glucose and you can measure hemoglobin A1C and you can measure cholesterol, okay? But if you put me in a hall with many people, including you, I don't know what's your glucose or cholesterol, but I know who's old and who's young. And this really attracted me very much to start to think about the basic biology of aging. The reason that I was hopeful is because it's clear, and I think now it's clear to anyone, at least when I say that there's a chronological age and a biological age. And you meet people who look amazing for their age and people who are like, what happened to them?
It's important to pause here for a moment to make this distinction clear, the difference between chronological and biological age. Chronological age counts birthdays, simply how many years since you were born, while biological age measures the wear and tear on the body shaped by genetics, environment, and lifestyle, and which is why two people of the same age can look and function very differently.
Right.
Which means there's some flexibility. I had an attending in the hospital when I was a resident that every time I presented the patient, he stopped me. I said, it's a 75-year-old woman. And he said, no, does she look older or younger than her age? Because it matters. You could have a 90-year-old person and with a little bit antibiotic and throw him out of the hospital, he'll be fine. And a 75-year-old woman, all systems are totally and it mattered. So I was looking then at, and I'm saying it because it'll come back in our discussion, at the model that really excited us all, the people in the field. You take animals, rats, mice, whatever, spiders, okay, Drosophila, and some of those animals, you let them eat whatever they want, and the others are calorically restricted. And the caloric restricted animals will live healthier and 40% longer. Okay. Like the first part of my career was, okay, when you do that, there's a lot of things are changing. Insulin goes down, adiponectin goes up, there's so what is it that is really important for their lifespan? Okay.
So as we discussed earlier, lifestyle interventions and doing exercise, just eating less are very effective. But as Dr. Barzilai regretfully mentions,
we knew that we cannot tell people to caloric restrict themselves. That's not going to work, okay? Very few. I mean, in diabetes clinic in the Bronx, I tell people diet. How many are dieting? 3%, It was not work. So we were looking for things that will imitate caloric restriction.
Let's pause for a second to understand this. Caloric restriction, just reducing the amount of calories consumed, of course, without malnutrition, has been shown to have widely beneficial effects on health span and lifespan. But now, could there be a pill for that?
And how would we measure that in a human study? There's a new branch of science that's addressing exactly these questions.
We call the field geroscience, and we're geroscientists. We have like 14 hallmarks of aging. How do we become a hallmark of aging? You show that something is wrong, and if you fix it, your animal, for example, lives healthier and longer. The interesting thing is those hallmarks are interconnected. You target one, you also affect the other.
And these geroscientists are now testing a whole bunch of medicines to see if they can prevent the ill effects of aging, including the drugs that we talked about, metformin, SGLT2 inhibitors, and an exciting new class of agents like GLP-1, which we will cover in our next episodes.
They were designed for diabetes. But lo and behold, in clinical studies, they do some other things. They prevent cardiovascular disease, they prevent kidney, they prevent Alzheimer, they prevent Parkinson, right? So if those things happen, that means we have drugs that are, we'll call that gerotherapeutics, that actually target the biology of aging.
In this episode, we are covering how it was discovered from the French lilac and Jean Stern's story. I feel it's stunning that a compound like that has such a dramatic effect on multiple different facets of human physiology.
Really, when I tell the metformin study, I tell about the fact that this French lilac was used actually for aging indication, for osteoarthritis, for prevention flu. They tried to prevent malaria with it. And it was kind of hijacked for diabetes because they noticed that people with diabetes had lower glucose, right? So then it became diabetes. And then in diabetes, they saw, in the UKPDS, they saw that, okay, metformin prevents cardiovascular disease. Sulfonylurea did not, insulin did not, but metformin prevented cardiovascular disease, that people with metformin have less cancers, that people with metformin have less cognitive dysfunctions.
And it will be highly exciting to see what this large clinical trial in people without diabetes, normal, healthy individuals taking metformin, will show.
TAME trial is going to start and expand with the help of the government. The important thing about the TAME, we went to the FDA and we said, if we have a drug that in a couple of years will change a constellation of outcomes of diseases, would you approve this drug? And they said yes. The trial is about showing that we're going to change the cluster of cardiovascular disease, cancer, cognitive decline, and mortality. And it was difficult for the FDA because we were agnostic to the disease.
Yeah.
And these conversations helped us underscore the fascinating aspect of how a humble plant extract from medieval Europe may be a powerful tool to fight the major scourges of humankind.
Yeah.
I appreciate what you're doing. This is really a good cause. And
Thank you.
and I'll check on you.
Thank you so much.
Bye,
you,
Ani.
thank you.
Bye-bye, guys.
As we conclude this episode, it is really incredible to reflect on how enormous an impact medicines inspired from nature have had on treating diseases and improving the human condition.
Exactly. When we look around us, in addition to the sugar-lowering medicines from the French lilac and the apple tree bark that we discussed in this episode, there are hundreds of millions of people whose lives have been extended or improved by statins from rice fungus for cholesterol, ACE inhibitors from snake venom for blood pressure, aspirin from the willow tree bark for prevention of heart attacks and strokes. These things treating and preventing complications that once seemed inevitable.
In fact, Pam, when we hear the term life support, we usually, think of pacemakers, dialysis machines, oxygen tanks, and sure enough, these are the visible mechanical forms of life support that we can see and touch and even hear. But
right.
what I have come to appreciate more now is these forms of what I call chemical life support, and just how many years of life and how many years of healthy living have actually been added because of these therapies.
That's so true. So, Ani, what's organic, natural, green, plant-based, and non-GMO? Most poisons.
That's funny. I have a poison joke too. If a poison is expired, is it more poisonous or is it less poisonous?
Love that one.
Well, in our next episodes, we go from talking about plant poisons to animal venoms and how the venom of a lizard from the Arizona desert inspired the GLP-1 revolution, launching drugs that not only transform diabetes care, but are poised to change society forever.
Wow. Those are big claims.
Big claims indeed. And we talk to scientists at the forefront of what may be the most earth-shaking drug discovery in a generation.
Stay with us for the Discovery Dialogues podcast.